Okay, so we have a number of questions for our panel. Why don't we start with the first one? And that is, probably would go to, I presume, Nina. Have you considered growing TILs ex vivo after vaccination? And then, I was going to finish. Oh, no, I know. I realized it was not going to be reinventing them, re-injecting them.
Absolutely.
We have audience participation here. But
Absolutely, and we're hoping actually in our neoantigen vaccine trials to, we're getting, obviously expanding T cells from these patients. We're going to be sequencing their TCRs, expanding the T cells. So we have a couple of things we'd like to do. One is have sort of a personalized expanded T cell population to give back to patients. In the shared neoantigen project, where plan is to sequence those TCRs and see if they're shared amongst patients, and that could be an adoptive cell therapy for the future. So that's another consideration. And as for TILs, we are now working on expanding, learning how to actually grow TILs and apply them into our vaccine center to do in the future.
okay. Question, I presume this should be for Miriam. What changes in the immune microenvironment occurred during MRD, minimal residual disease, or in the relapse phase? What can we learn in those settings?
So we have not focused yet enough, I think, on MRD, and we have not also focused so much on relapses. Our major effort has been to really map tumor lesions that have been untreated for many reasons. We wanted to start to establish these baseline atlases that are going to really help us understand the disease better. It started to be a little bit of a mess when the lesions have been exposed to multiple therapy. Okay, so now there are several efforts to now start understanding MRD. In fact, with the myeloma group, we are developing an MRD test to be able to really and hopefully understand these minimal residual cells and understand the biology of these cells. There's a lot to learn. With Julio, we are very interested in also looking at this compartment as deeply as possible and looking also at the microenvironment where these rare cells that remain, in fact, engraft, and why do they have this biology, and whether the tissue is, in fact, impacting the biology. So I will argue that absolutely we should call it, apparently not the immune monitoring, because we go beyond the immune monitoring. We will also look at the cells, at the tumor cells itself, at the stromal cells, and at the tissue. So this monitoring and this assay that we develop are indeed, I think, going to be very informative to understand disease course.
Great. So another question, this would be for Nina, and basically there have been two approaches to define neoantigens. One is the predictive approach, and the other has been more physical, and that is eluting antigens from the MHC. Why aren't you doing that? And which do you think is going to wind up giving us the best neoantigens?
Well, the first question is easier to answer than the second. We'd very much like to do the first, but it really requires very sensitive mass spec to do that, and a lot of tumor material to do the elution. You need about 200 million cells to do this, and it's something we'd love to do in the future, obviously, in order to correlate. But there are many groups that are doing this at the moment, and there's a big need to do it because we don't really have a very good sort of library of epitopes that bind, for example, HLA class 1B or C. So these are endeavors that need to happen, and we'd love to be part of that initiative eventually. So the second question is going to be interesting because we really don't know. We need more data, essentially. We need data understanding what we can find and predict from our genome sequencing and what's actually eluted off a tumor cell, because there are lots of steps in between. There's the immunoproteasome, which will affect processing. We now know then the proteasome, there's splicing of peptides, which you will not be able to predict really. You need to elute it off of the class 1 in order to understand how much splicing occurs. And there have been estimates that maybe 30% of peptides arise from spliced peptides. And then the other thing that comes up is the concept of these phosphopeptides, so citrullonated peptides. So the modifications, once peptides are even made, that you wouldn't be able to predict necessarily. So there's a whole new class of epitopes that could be very important and are actually being tested by colleagues in the clinic as well. So this is something I think we really need to do, and I wish we could do it right away.
But I think the point you make, which has been found, that is, you can now have protein splicing to come up with peptides that you might miss, as you kind of have to depend upon Ben to define the neoantigens, whether your algorithms can pick those up or not.
Well, I mean, I wasn't the person who did this work, several of whom are here. I think that, the field is driven by computational tools that are useful but limited and that have a lot of room for improvement, and that's part of the whole, collective project. The sort of dichotomy that was just set up where there's computational approaches and mass spec-based approaches, the computational approaches use mass spec data to try to get better. Several people in this room have been a part of that. And so, and yet with these limited tools, as Nina's and the OpenVex work shows, you seem to get something. So it tells you something also about how to compare.
And we'll find out, right?
But I think computationally, it also tells you something about how to benchmark new approaches as they come online, which is that these tools, despite all of their limitations, seem to give you biological information.
So we have a little more, about a minute more, but an interesting question from Marta, and that is there data on evolutionary fitness now, but of metastatic tumors? And is there some way that we can see that fitness applied to metastatic tumors versus the naive tumors?
Yeah, I think this is a very interesting problem from modeling point of view. How, what we would expect also to see under this model in terms of metastasis. For instance, the clones that have high, immunogenic antigens should not really go to metastase. And we can also treat this data as some kind of longitudinal data that could actually give us better ways to validate and then improve this models, because this is actually what we want to really see how tumor will evolve and what's going to happen, and metastasis is just a perfect setting.
That clearly is very interesting, at least the immunologists up here. I think we're down to 17 seconds, so I think we probably should, can get in 2 or 3 more questions, but I think maybe we should stop here and thank the panel.